Welcome to Pharma Drama, the channel where we look at the science of healthcare and healthcare products. As you might know, I make my UCL lectures freely available, and this is a lecture I deliver to our M.Pharm and MSc students. So if you're one of our UCL students, welcome, you've clicked on the right link. And if you're not at UCL, also welcome. You can watch the lectures without fear of an exam at the end. As with all of my lectures, there is a set of PowerPoint slides to accompany the videos. If you are at UCL, you will have access to the slides. If you're not at UCL but would like a copy, just send me an email. I recommend that you have the slides open as you watch the videos, and as I progress through, I will put the slide I'm talking about on the screen so you know what I'm referring to. Today we're going to talk about a new type of manufacturing paradigm that has the potential to change the way we design and deliver medicines: 3D printing. I'm sure you've heard of 3D printing or read about some of the amazing things that have been 3D printed: organs, body parts, guns. Although I have to be clear, in the UK at least, it is absolutely illegal to print any part of a gun, so don't even go there. While we are not quite at the level of printing new body parts in pharmaceutics yet, I can tell you that 3D printing is revolutionising the types of drug delivery systems we can make, and is allowing us to personalise medicines to individual patients. In this lecture, I will introduce the types of 3D printing that are most widely used in the design of medicines and look at some applications of each. We will have regular coffee breaks through the lecture, and I see no reason not to start with a coffee, and I recommend you get a drink too. Then when you're ready, press print and we'll make a start. As ever, let's start with today's learning objectives, which are shown on the screen in front of you. By the end of the lecture, you should appreciate what types of drugs the pharma industry has to formulate and the challenges it faces in delivering them, particularly in respect of biological drugs and personalised medicines. You should also be able to describe the operating principles of a number of printing technologies: powder bed, fused filament, stereolithographic, selective laser sintering, and gel printing. In all cases, you should be able to discuss how these printers work, what type of material they can print, and their advantages and limitations. We will also discuss some pharmaceutical applications of each, so you should also be able to give examples of their medical use. In each case, I will provide links to research papers that you can read to see the original research. Spoiler alert, most are from my own research group. And of course, you are free to find your own research papers because printing medicines is a very big field right now. Right, let's start the lecture by considering what types of drugs the pharma industry needs to formulate. On the screen in front of you are the structures of two classic drug molecules, aspirin and paracetamol. As well as being among the oldest drugs to have been developed, they are also typical of the types of molecules that have been used as drugs, in that they are low molecular weight organic compounds. By low molecular weight, I mean under 500. You might remember this is one of Lipinski's rule of 5, a useful guide to the likely potential of a molecule to be used as a drug. Such molecules are easy to characterise, as they are usually white crystalline powders and often attainable in polymorphic forms, each with specific melting points. The first property of such molecules that is measured is solubility, and the second is usually partition coefficient, which is related to permeability. I have posted a couple of short videos on solubility and partitioning should you be interested in these concepts, and the links are in the description below. With these initial data, an early indication on the likely developability of a compound can be made using the Biopharmaceutical Classification System, or BCS. As you can see on the screen, this puts molecules into one of 4 categories, based on solubility and partitioning. BCS Class 1 compounds have high solubility and are highly permeable. These should be very easy to formulate. BCS Class 2 compounds are highly permeable but have low solubility. In this case, clever formulation can often overcome the solubility issue, and so these molecules are also relatively easy to formulate. BCS Class 3 and 4 compounds present more of a challenge, however. Class 3 compounds have low permeability but good solubility. Thus, if the solubility is high, enough of the drug can probably be absorbed to elicit a therapeutic effect. So I have coloured this category red and green. Class 4 compounds, though, have poor everything. Even if clever formulation can improve their poor solubility, they are still poorly absorbed. Thus, Class 4 compounds are always a challenge to deliver, and so I've coloured these red. Once a compound has been selected for development, the chances are it will end up formulated as either a tablet or a capsule. Principally because, at least in the US and the UK, this is the dosage form that most patients are familiar with. Since they contain the drug in its solid state, which is the most stable state, they also have the longest shelf lives. So this is how the pharma industry has been set up for many years, formulating small molecular weight organic compounds that are usually attainable in a crystalline form in tablets and capsules. How does this setup compare with the drug products that the pharma industry actually sells? On the screen in front of you is a table showing the best-selling drugs from 2010. Ranked by sales volume, of course, the pharma industry is money-driven. 3 of the products have asterisks. Do you know what this signifies? I'll give you a moment to think about that while I have some coffee. How to think? The answer is these drugs are biologicals, monoclonal antibodies in this case. The remaining 7 are small molecular weight organic compounds, which fits with the model of the pharma industry I've just discussed. Now let's look at the same data from 2020. Quite a change, I think you'll agree. Now 7 of the top 10 are biologicals and only 3 are small molecular weight organic compounds. This change in the pipeline of drugs being developed has profound consequences for the pharma industry, and it cannot keep the current mass production tablet capsule model for much longer. Why? Well, for one thing, production of tablets and capsules is only economic in bulk scale. Each production run must yield hundreds of thousands of tablets or capsules, and each must have exactly the same dose. Tablets and capsules are available in different dosages, of course, but usually the range is quite limited. The limited dose range becomes a problem when a drug has what is called a narrow therapeutic index. That is, the dose required for the patient is quite exact. In this case, patients may have to divide tablets or take multiple tablets or capsules to get their required dose. Warfarin is a good example of this. This problem is becoming more common because many drugs are now designed on computers, and so their affinity to biological targets is very high. Also, where patients require multiple medications, they must take multiple tablets or capsules each day. And I am sure you are aware, patient non-adherence to medication is one of the main reasons why conditions don't improve. A good example is provided by blood pressure medications. You probably know there are many classes of antihypertensives, and if a patient takes only one, the chance of successfully controlling blood pressure is low. A recent study in The Lancet showed that where patients took 4 classes of antihypertensives daily, it was possible to achieve nearly complete control of blood pressure. Of course, this means patients taking 4 tablets in the morning and 4 in the evening, which is going to lead inevitably to poor compliance. A better option would be a polypill containing all 4 drugs in the right doses. More on this later. An additional complication is the increasing number of biological drugs, which have their own formulation requirements. Being proteins, biological drugs are very difficult to administer orally because they will be digested before being absorbed. Also, they often have a biological structure, secondary, tertiary or quaternary, that is required for activity and can only be maintained while in solution. And in many cases, very good control of solution factors such as temperature, ionic strength and pH is needed. For these reasons, biologicals are frequently formulated as solutions for IV injection. While this guarantees 100% bioavailability, it is not great for patient compliance. This leads to the question, is there an alternative manufacturing paradigm that would allow the pharma industry to adapt to meet some of these challenges. A method of manufacturing that would allow the formulation of biological drugs, the production of small batches with almost limitless variability in dose, and the option to make medicines tailored to the needs of individual patients. I believe there is, and it's 3D printing. Before we take a coffee break, let me introduce what 3D printing actually is. 3D printing is often referred to as additive manufacturing, and while I don't think this term is quite as catchy, it does help to understand how the process of 3D printing works. Think first about conventional manufacturing. Since humans invented tools many thousands of years ago, we have made things by starting with a block of material, like wood or metal, and removing material to fashion that object. In this sense, we can think of conventional manufacturing as subtractive. We start with a block of something solid and we remove material from it to make our object. 3D printing turns this paradigm on its head. Starting with nothing, we add material layer by layer to create our final object. And this is why the term additive manufacturing is used. All 3D printers work by depositing some material, I call it a feedstock material, and setting it solid in some way. The reason there are so many types of 3D printer is because each type builds objects out of a different material and or uses a different process to set the material solid, as we shall see in a moment. We will start to look at specific 3D printing technologies after the break. But before that, you might ask me, how does a 3D printer know what to print? And that is a good question. Like any printer, a set of instructions is sent by a computer telling it what to print. To make a three-dimensional object, a 3D printer requires information on what has to be printed to make each layer of an object. To do this, once the object has been designed, it must be sliced into a series of horizontal layers. The easiest way to do this is to design the object in CAD software. There are loads of software options available that you can use to design three-dimensional objects. We tend to use Tinkercad. A link is given below because it is web-based and free. But with a UCL account, you can also download AutoCAD. If you don't want to design your own objects, then you can download one someone else has designed. The biggest resource is Thingiverse, and again, you will find a link for that below. When you press print, the software automatically slices your object into horizontal layers and sends the information for each to the printer. The printer then deposits material onto a build plate layer by layer to create the final object. 3D printers can make objects out of metal, ceramic, plastic, resin, gel, powder, foods, and probably more. The common feature that links all 3D printers is that they need to be able to dispense the material they are printing and then set it solid. Why do 3D printers only print solid objects? I also hear you ask. The answer is that there is no point trying to print a liquid or gas. Imagine the mess. So the object must be solid, or at least semi-solid if a resin or gel is printed. But how can a printer dispense something that is a solid? The answer is that it doesn't. The really clever part of 3D printing is in how the printer takes a raw material and processes it to become a solid. When we come back, we will look at the most common 3D printing types that are useful for printing medicines: powder bed, fused filament, stereolithographic, selective laser sintering, and gel printing. So, with that to look forward to, have a break, get a new drink, and I'll see you in a bit.